Okay, meiosis. This one is going to be a doozy. So just get comfy, get your snacks, get your Cheetos, get your Diet Dr. Peppers, and just hang out. Here we go. So meiosis. Hopefully this first part is going to be a bit of a review for you guys. Meiosis's job is the gametes. So we know that the body cells, all of our other cells are diploid. There's 2 copies in everything. There's 2 copies in our stomach cells, our hair cells, our fingernail cells. But with our sperm and or egg cells, there's just going to be haploid. It's 1 copy. Because remember, the goal of those cells is to fertilize another, or to engage in fertilization with another cell of the same or different to make a new person. All right? So it's haploid. So you've got a haploid sperm and a haploid egg making a diploid you in the miracle of life. All right, so we call it sexual reproduction. What that means is there's 2 parents, right? Asexual is when you have 1 parent and it just creates a new thing, a new entity. We see this especially in like bacteria and protists and single-celled organisms. We see asexual reproduction. All of the higher level living things, we're going to see sexual reproduction, 2 parents. So that means 2 divisions. Funny. We call that meiosis I and meiosis II. Clever names. All right, let's get into it. Like I said, the goal here is the formation of the zygote, which is essentially the fertilized sperm and egg. You have the sperm has 23 chromosomes. The egg has 23 chromosomes. They combine to make a little zygote, which then turns into you. And that's 46. So that's where we get this idea of haploid. Haploid plus haploid equals diploid. And that's important to note, of course, because that's how we get new people. We get new-looking people. We get diversity in our gene pool based on half of the chromosomes coming from your mom and half of them coming from your father. Now, if you're like a hill person from, where hill people live, and you were married to your cousin or your brother or your sister, whatever, however it works, that's where you see problems because, for many reasons, but that's where you see there's not a whole lot of genetic variance. So you see kind of some problems with people who interbreed. I'm not saying that there are lots of instances of these. It's just like, for example, right? Anyway, so interphase 1. So I'm going to take it real slow on these first couple steps. Then I'm going to kind of yada yada the last couple steps, because the first couple steps are going to be more important. So it's very similar to mitosis. You're having chromosomes replicate. Chromosomes only replicate in interphase 1. There really is no interphase before the second division in meiosis. So you have what you normally have. You've got your chromatids, ba-boom, 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 chromatids, yeesh, and your centromere that holds it all together, okay? Interphase 1, very similar to mitosis. Now remember, this is not happening in your other cells. It's only happening in your ovaries and your testes, depending on which one you have. And if that's a question for you, then, you can talk to your doctor, your counselor, go on the internet. Okay, so cell division is going to reduce that number of chromosomes by 1/2. That's our goal here, because we only replicate once. The goal there is to reduce that chromosome number. So there's 4 phases in meiosis I: prophase, metaphase, anaphase, telophase. You remember the amoeba sisters were like, PMAT, get it? And you're like, yeah, this is great. Thank you, amoeba sisters, for giving me such an engaging and exciting learning experience. That's what you meant to say. So yeah, so you've got the chromosome, or meiosis I, 4 phases, just like mitosis, just like meiosis II. Okay, there's a pattern here. So here's the money. Prophase 1 is the money. This is where the magic happens, okay? This is where you're having the tetrads formed, okay? And remember, the tetrads are going to be 2, oh boy. So say this is from mom. This is your chromosome from mom. and say this one's your one from dad. We'll color that one in. Okay? So that's your tetrad right there. And that's where you have crossing over, okay? So you have this exchange of genetic material here that makes you unique, okay? So this is why this is so important, because crossing over happens in prophase 1, and that's why you're not an identical clone to your sisters and your brothers. Yeah. So this would be like the sister chromosome. Like, so if this is our mom's chromosome, this is a sister chromosome, or a sister chromatid. This is a non-sister chromatid. So that's all that just means. Okay, so this was supposed to be a really awesome drawing that never happened. So we'll just do this. Yay, that's your dad chromosome. This is mom. This is basically what I just said. And then they come together to form the tetrad. Tet meaning 4, okay? So there's going to be 4 chromatids, 1 from each. That's all that's saying. Okay, so your homologous chromosomes, right? Homo meaning same, logos meaning place. So they're going to be similar in shape and size. They're going to be the same number of, they're going to be the same chromosome number, 21, 22, 4, okay? So they carry genes that control the same inherited traits: eye color, hair color, height, skin, melanin, amount of melanin in your skin, etc. And then they each have the gene at the same location. So if you have, for example, if on, let's say chromosome 4, if this controls eye color, it's going to control eye color on everybody's chromosome in that same location. Now, what this gene codes for is going to be different, but the location is going to be the same. As we know, we've got 23 pairs of chromosomes. We have 22 pairs of autosomes. These are your body cells. And then we have 1 pair of your sex chromosomes. This is your X and your Y, or your double X. Oh gosh, so this was supposed to happen too. Okay, so I'll just draw this, draw this. There we go. So this is just basically an example of crossing over. So I'll just draw this. So what's going to happen is you've got these two, they're hanging out. Maybe the eye color locus is here, okay? And the eye color locus is here. And what you're going to have is you might have crossing over. So you might have a situation where you've got your father's chromosome. I'll put the hair color down here. So you've got your father and your mother chromosome. You might have a situation where, like, this would be your mom's, and now you've got some of your dad's DNA there. And that's the same with the hair color. You might have, we'll leave that blank to represent the mother's. So you basically have switching of DNA. So your DNA gets switched, which is good because that supports variation, right? My eye color is, my eyes are greenish. My dad's and my younger brothers are like a hazel, like a dark green brown, and my mom and my brother had clear blue eyes. So, like, whoopsies, sometimes that happens, sometimes with crossing over and with meiosis, you get a shift of your genes, or you get your genes in a way that make for new traits. I also am the only one in my family that has the inability to tan, horrible eyesight, and I went prematurely gray quicker than my younger brother did, but he went gray too. So I'm a walking, talking recessive gene. Wah, wah. You know this. Each one of us have 23 sets of homologous chromosomes, and then you've got this guy. This would be a lady, because usually this is XX. XY looks kind of like X. This is X and this is Y. Rank. So yeah, so that's a karyotype. We know what that looks like. That's a map of your chromosomes. Again, crossing over occurs between non-sister chromatids. So they're in homologous chromosomes, but they're crossing over, so from different parents, essentially. Okay, so what happens is the DNA breaks, so the chromatids break and reattach. And that's where that DNA switches. There's a whole bunch of enzymes that code for this. This isn't necessarily, this is controlled by enzymes. It's not like all willy-nilly that DNA is like flying all over the place and new DNA is being made and there's a fear that you could grow. You would have a baby with like, a shark fin on the back just because, DNA went crazy. That's not the case. It is somewhat controlled, but that's what crossing over is. So you see this, guys, you see this on your handouts, I know. So we know what this looks like. We know how this works. You're making me crazy. That's that, tetrad, variation. So these are going to be non-sister chromatids. So here we go. Whoopsie. And then you've got this, and then you've got touching. Okay? And then you have that switch, what we saw before. So this is where the switching would happen. Okay? Cool. All right. So that's basically what we're talking about here. So that's, like I said, prophase accounts for 90% of what's happening in meiosis. But there's still a ton of really important stuff that's happening. So let's kind of go through this. Let me just make sure I have everything that we need to know. Great. So let's kind of go through and find out and just kind of whip through what's going on in these other steps. So prophase 1 is the money. This guy is the shortest phase. Metaphase 2 is the shortest phase. Okay. So you have these guys lining up against the equator right here, and they're about to split. So the thing that's different, though, if you look, you've got the homologous pairs separate. So in mitosis, you have the chromatids separate. Not so. The entire chromosome separate. Okay? So in this case, the centromeres are still attached. You've not broken the centromeres. So here's something else too. Depending on how they line up at the equator, that determines a lot. Because the combination you have down here in your gametes depends on how they all line up and separate here. So where they metaphase is important because it kind of like, they independently, it's called independent assortment, where they line up against the equator and they separate randomly. So you don't, we don't really know. There's a chance of probability and there's some unknown there in terms of what sperm and what egg, what chromosomes are going to be in each gamete. Okay, so telophase 2, boom, cytokinesis, they break. Okay, here's the thing. No interphase before prophase 2. There is no replication. Okay, so after telophase, we have kind of these two new cells after cytokinesis. This is very similar to mitosis. Meiosis II is very similar to mitosis. You see them lining up against the equator. You see the spindles attaching to the centromeres. You see them separating. But again, instead of, because this is happening twice for two different cells, you end up with 4 cells. You get 4 cells and they're genetically different from their parents. That spells different. Okay, so if you look, you can even kind of see how there's like there's an orange and red chromosome there, there's like a purple chromosome here, there's a purple and blue chromosome here. Totally rando, right? So imagine that these were sperm, okay? If these were sperm, now whichever guy wins, and what I mean is like penetrates that, swims up the birth canal, which believe you me is no easy journey, swims up the birth canal through the uterus into the fallopian tubes, right? And fertilizes an egg. And what has to happen is the sperm has to actually, there's an enzyme at the end of it where it has to dissolve the outer part of the egg cell to inject the DNA, the genetic material. So say that happens, right? Say winner, chicken dinner. Whichever one wins, it's completely up. In many ways, it's random to see what DNA will be passed on to the next generation. So, it's the miracle of life, guys. So that's what I got. Hopefully I've cleared this up more than I have not cleared it up. Hopefully my Hill People reference was taken in the spirit in which it was intended. And bring your questions tomorrow and we'll go over them. Sweet.